Good day, everyone. Today, we'll discuss about the two ways in sanitizing. Sanitizing, the primary reason for the application of effective sanitizing procedures is to destroy those disease organisms which may be present on equipment or utensils after cleaning, and thus prevent the transfer of such organisms to the ultimate consumer. In addition, sanitizing procedures may prevent spoilage of foods or prevent the interference of the microorganisms in various industrial processes which depend on pure cultures.
Two ways in sanitizing. 1. Chemical. Accomplished by immersing or wiping with sanitizing solution and allowing the solution to remain in contact in a solution for a specified amount of time. 2. Heat. Exposing the equipment to high heat for an adequate length of time. This is done manually by immersing equipment into water at 171 degrees Celsius Fahrenheit to 191 degrees Celsius Fahrenheit for at least 30 seconds. Sanitizing equipment, heat, and chemical. Heat composed of steam, hot water, and radiation, while chemical composed of chlorine, iodine, and quartz. Heat sanitation, steam, exposing the utensils to steam 170 Fahrenheit for 15 minutes. Hot water, manual washing, 171 Fahrenheit for 30 second machine, 180 Fahrenheit for 20 seconds. Radiation. Ultraviolet radiation can be used to sanitize but is not used in most food service establishments. Its major application is in the packaging areas of food processing facilities. The contact time should be at least 2 minutes. It only destroys those microorganisms that are in direct contact with the rays of light. Different types of sanitizing solutions. Antiseptic, an agent used against sepsis or putrefaction in connection with human beings or animals. Disinfectant, an agent that is applied to inanimate objects. It does not necessarily kill all organisms. Sanitizer, an agent that reduces the microbiological contamination to levels conforming to local health organizations. Germicide, an agent that destroys microorganisms. Bactericide, an agent that causes the death of a specific group of microorganisms. Bacteriostat, an agent that prevents the growth of a specific group of microorganisms. Sanitization, the process of reducing microbiological contamination to a level that is acceptable to local health regulations. Sterilization, the process of destroying all microorganisms. Three most common chemical sanitizers. Chlorine. It is effective in hard water, but it is inactivated in hot water above 120 degrees Celsius Fahrenheit. Advantages. Effective on a wide variety of bacteria. Highly effective. Not affected by hard water salts. Generally inexpensive. Disadvantages. Corrosive, irritating to the skin. Effectiveness decreases with increasing pH of solutions, deteriorates during storage, and when exposed to light, dissipates rapidly, loses activity in the presence of organic matter.
Rule for thumb mixture for chlorine sanitizing solution. 50 parts per million solution. 1 tablespoon of 5% commercial chlorine solution. Mix with 4 gallons of water and sanitize the solution for 7 seconds. Temperature of 75 degrees Fahrenheit to 115 degrees Fahrenheit. Used to sanitize food thermometers. 100 parts per million solution. 1 tablespoon mix with 2 gallons of water. 200 parts per million solution. 1 tablespoon mix with 1 gallon of water. Iodine, effective at low concentrations, less effective than chlorine, becomes corrosive to some metals at temperature above 120 degrees Fahrenheit, more expensive than chlorine. Advantages: forms brown color that is indicative of the germicidal strength, not affected by hard water salts, less irritating to the skin than is chlorine, active against a wide variety of non-spore-forming bacteria, and not lost as rapidly as chlorine in the presence of organic matter. Disadvantages: bactericidal effectiveness decreases greatly with an increase in pH. Most active at pH 3.0 and very low acting at pH 7.0, less effective against bacterial spores and bacteriophage than is chlorine. Should not be used at temperatures greater than 120 degrees Fahrenheit and may discolor equipment and surfaces. Quaternary ammonium compounds, or QUATs, not as quickly inactivated by soil as chlorine, remain active for a short period of time after it has been dried. Non-corrosive, non-irritating to skin, works in most temperature and pH ranges, leaves a film on surface, does not kill certain types of microorganisms, hard water reduce effectiveness. 3-sink dishwashing method is widely known and used for food establishments such as restaurants and bars. Wash. These steps uses a detergent solution to remove dirt from the surface. The detergent must be approved for food use and able to dissolve and remove dirt. Rinse. This is the final washing step. It removes any dissolved substances or detergent residue remaining on the surface, which could decrease the effectiveness of the sanitizing solution. Sanitize. These steps make sure all surfaces are entirely free of bacteria and biofilms.
Sanitizing large equipment. Step 1. Unplug. Step 2. Remove loose food particles. Step 3. Wash, rinse, and sanitize any removable parts. Step 4. Wash the remaining food contact surfaces and rinse with clean water. Step 5. Clean surfaces. Lastly, Step 6. Re-sanitize the external food contact surfaces. Here are the factors affecting the action of chemical sanitizers. Contact of the sanitizer. In order for a chemical to react with microorganisms, it must achieve intimate contact. Selectivity of the sanitizer. Certain sanitizers are non-selective in their ability to destroy a wide variety of microorganisms, while others describe a degree of selectivity. Concentration of the sanitizer. In general, the more concentrated a sanitizer, the more rapid and certain its action. Increases in concentration are usually related to exponential increases in effectiveness until a certain point where it accomplishes less noticeable effectiveness. Temperature of solution. All of the common sanitizers increase in activity as the solution temperature increases. This is partly based on the principle that chemical reactions in general are speeded up by raising the temperature. However, a higher temperature also generally lowers surface tension. increases pH, decreases viscosity, and affects other changes which may enhance its germicidal action. It should be noted that chlorine compounds are more corrosive at high temperatures, and iodine tends to sublime at temperature above 120 degrees Fahrenheit. pH of solution. The pH of the solution exerts a very pronounced influence on most sanitizers. Quaternary compounds present a varied reaction to pH depending on the type of organisms being destroyed. Chlorine and iodophore generally decrease in effectiveness with an increase in pH. Time of exposure. Sufficient time must be allowed for whatever chemical reactions that occur to destroy the microorganism. That required time will not only depend on the preceding factors but on microorganism populations and the populations of cells having varied susceptibility to the sanitizer due to cell age, spore formation, and other physiological factors of the microorganisms.